My name is Dr. Jamie Caroon, and I am the medical director at the Center for Medical Cannabis Education in San Diego. Today, I am going to present some ideas on the dosing and administration of cannabis, more specifically, the dosing and administration of cannabinoids for medical purposes with a focus on THC and CBD. In this presentation, I am going to attempt to characterize some of the very complex dynamics around dosing and administration of cannabinoids as medical therapies. We're going to talk about the pharmacokinetic differences between various methods of administration and how the method of administration largely determines the pharmacokinetics. We're going to look at the dose-response relationship to get a sense for whether there is a linear relationship. Is more better? Or is it a more sigmoidal shaped curve, an S-shaped curve where there's no incremental benefit for a higher dose, which is the type of dose response curve we see a lot with pharmacological medications. We're going to discuss the contraindications and drug interactions so that we can be focused on safe application. And then at the very end, we'll talk about a method of dose titration that is based upon tolerability before clinical efficacy. So with that, let's get into it. One of the challenges of using cannabinoids in clinical practice is the lack of clinical resources to consult when you don't know what to do. Let's look at how we might approach this problem if we were talking about an FDA-approved drug. So we're looking at a 53-year-old male with essential hypertension that has not been controlled with intensive lifestyle modification. You decide to initiate pharmacological antihypertensive therapy. You're not sure what drug to use and what dose. What would you do? You would either consult with a colleague or use a clinical resource like UpToDate. So if we were to use UpToDate, it would likely tell you to go with a thiazide diuretic, probably chlorthalidone, and it would give you guidance on initial dosing, 25 milligrams once daily. Wouldn't it be nice if we had resources like this when we were thinking about using cannabinoids? Well, we actually do have some information like this. There are two FDA-approved synthetic or semi-synthetic versions of THC, dronabinol and nabilone. Dronabinol was first approved in 1985 to treat chemotherapy-induced nausea and vomiting. Its indication for use was extended in 1992 to include patients with HIV and AIDS who were suffering from anorexia and cachexia. It can also be used off-label to treat obstructive sleep apnea and chronic pain. Dronabinol is the same chemical formula and the same chemical structure as naturally synthesized THC. It's a Schedule III controlled substance. Naturally synthesized THC is a Schedule I controlled substance. Nabilone is Schedule II. It is a different chemical formula, different chemical structure. It's got a greater affinity for the CB1 receptor and greater pharmacodynamics, so higher potential for abuse. Both of these drugs are FDA approved for the same indications, and so nabilone covers the same conditions or clinical situations. Nabilone was also approved to treat fibromyalgia in other countries, and it was approved in 2006. Both of these drugs are single molecules. They're synthetic and semi-synthetic versions of THC. They are both manufactured according to pharmaceutical-grade GMPs, and they are also listed in UpToDate. So if you wanted to know what dose of dronabinol to use for a patient with anorexia secondary to HIV and AIDS, it would tell you 2.5 milligrams twice daily. And then finally, there is Epidiolex, which is a newly approved cannabinoid as of June 2018. Epidiolex as a drug product is 99% naturally occurring CBD extracted from marijuana. It's administered as an oral solution. Both dronabinol and nabilone are administered as capsules. Epidiolex was approved to treat two rare seizure disorders, Lennox-Gastaut syndrome and Dravet syndrome. Epidiolex is a Schedule V controlled substance. The scheduling of Epidiolex by the DEA represents the first time that the DEA acknowledged that marijuana had any currently accepted medical use. If we wanted to know what dose to start with using Epidiolex, we could look at the drug monograph where it would tell us that 2.5 milligrams per kilogram of Epidiolex, which is 99% CBD, should be taken twice daily. And you can even find some guidance on using some of these drugs off-label by looking at clinical trials. Here's a study that found that dronabinol had an additive pain-relieving effect among patients taking opioids for chronic pain. Dronabinol is not approved to treat chronic pain. So by way of review, we have at least a place to start for oral dosing of isolated THC and isolated CBD for a number of symptoms or medical conditions. But these drugs are not commonly prescribed, and Epidiolex is so new we don't have any prescribing data on it yet. It is popular to believe that the effects, both the therapeutic and the adverse effects, are different when you are comparing a synthetic to a natural compound, and when you are comparing an isolate to a whole plant extract. And we will touch a little bit on this in the slides ahead. The entourage effect, which you have probably heard of in the world of cannabis, is used in cannabis lore to describe botanical synergy, which is a concept that describes how secondary compounds in a formula may modulate the pharmacokinetics and the pharmacodynamics of each other, or perhaps a primary compound or ingredient. There's one other thing I just wanted to point out in this slide before we move on, and that is the highlighted sentence, which says that there were no differences that were found between the 20 milligram dose and the 10 milligram dose. We're also going to talk about this idea that more may not necessarily be better, and that the dose response curve is not linear. Okay, let's take a step back now and just talk generally about factors that go into dosing with an eye towards medical cannabis as it is typically being used today, which is in its form either as a plant or as a plant extract or concentrate. Demographic factors are obviously very important. A patient's age at either end of the age spectrum is probably important, for example. Health status is also key. Pregnancy is usually a contraindication. Comorbidities could have an impact on liver function, kidney function, or GI function. A history of cannabis use can give you some information about how they are likely to respond to the psychotropic effects. Cannabis is very safe, but tolerability is really the issue in the beginning, especially with an initial dose. It's important that patients don't have an uncomfortable first experience. And dosing for tolerability is different than dosing for efficacy. The method of administration determines the pharmacokinetics, which determines the effects. And dosing may be different if you are using a different method of administration. It likely is. Dosing will probably be different if you are administering an isolated compound versus a whole plant extract. Potency is the percentage of THC or CBD in a given sample. It is not dose. THC is much more potent than CBD is on a per milligram basis. And ratios are also not dose. These are typically expressed as CBD to THC. Often you'll hear a 2-to-1 or 8-to-1, something like that. And obviously this is not dose because a 2-to-1 could be 10 milligrams of CBD and 5 milligrams of THC, or it could be 100 milligrams of CBD and 50 milligrams of THC. Both of those are a 2-to-1. And then finally, tolerance is important. It is principally influenced by chronic usage, which results in downregulation of cannabinoid receptors. And tolerance is a component of this concept of endocannabinoid tone. To a large degree, the method of administration determines the pharmacokinetics. Pharmacokinetics can be thought of as what the body does to the drug. Pharmacokinetics can be broken down into 4 principal components: absorption of the drug, distribution, metabolism, and then finally excretion of the drug. Most specifically, the method of administration determines the absorption. And inhalation and ingestion are the most common methods, and pharmacokinetic studies show that the bioavailability of cannabinoids is greater through the respiratory tract than it is through the gastrointestinal tract. Now, there's a lot of variability in these studies, as there is in human beings, and these are just estimates, but it's thought that roughly 10 to 60% of the dose of THC administered via inhalation can be measured in the blood thereafter. And for ingestion, the peak level is even lower, roughly 6 to 30%. And these estimates are comparable for CBD, but most of the studies are looking at THC. These phytocannabinoids are lipid-soluble, oxygen-containing hydrocarbons. So they have very poor solubility in the GI tract, and as a result, they are not very bioavailable. During first-pass metabolism in the liver, delta-9 THC is metabolized mainly to 11-hydroxy-THC and then to 11-carboxy-THC. 11-hydroxy-THC is more potent than delta-9, and 11-carboxy-THC is basically inert. And the metabolism is mediated by the cytochrome P450 enzyme complex, which metabolizes, they say, 60% of all drugs, I think both pharmacological and illicit drugs. The subunits are 2C9, 2C19, and 3A4 for the most part. And as far as excretion is concerned, slightly more than half of the THC that is administered will be excreted in feces, and roughly 20% will be eliminated in urine. We're going to talk more about pharmacokinetics in a few slides. Now let's look at the pharmacology of THC and CBD. THC is a partial agonist of the CB1 receptor. It binds to the orthosteric site on the receptor, which means that when it's bound, THC initiates a biochemical cascade inside the cell that results in something happening, a gene being turned on, a gene being turned off, an enzyme being activated, a protein being synthesized, whatever the case may be. Cannabinoid receptors are G-protein coupled receptors. In fact, these are the most concentrated, the most densely populated receptors in the human central nervous system. CBD is often described as a functional antagonist of the CB1 receptor. It binds to the allosteric site of the receptor, which is different than the orthosteric site. When bound, CBD changes the conformation or the shape of the receptor, which decreases the affinity of THC at the orthosteric site. And this is known as allosteric modulation because CBD decreases the affinity of THC. And as a result, it's a negative allosteric modulator. If it increased the affinity of THC, it would be considered a positive allosteric modulator. Interestingly, CBD and THC, but more so CBD, has an affinity for a variety of different other receptors in the central nervous system, including serotonin receptors, GABA receptors, glycine, adenosine receptors, etc. And CBD acts as a positive allosteric modulator for a GABA receptor, and THC acts as a positive allosteric modulator for at least one of the three opioid receptors. From a dosing perspective, this allosteric modulation is important because the dose of THC may produce a different subjective experience for the patient when CBD is administered concurrently, or even if it's administered before THC, because it may change the shape of the receptor, and as a result, it may change the pharmacodynamics inside the cell. The pharmacodynamics, or the intracellular response, is different depending on the agonist. This is called agonist trafficking, and it's very important. THC inhibits adenylate cyclase. It also inhibits the opening of calcium channels, where anandamide, which is a different agonist at the CB1 receptor, only inhibits adenylate cyclase. These are two synthetic cannabinoids that are full agonists at the receptor and have different effects than each other and as THC and anandamide. The point is that these are very complex systems, and we need a lot more information before we can really understand what's going on here. Let's talk a bit about safety. Human beings have been using cannabis for thousands of years without an overdose death being reported. And even the DEA supports this claim. Meanwhile, in 2016, over 42,000 Americans died from overdoses associated with prescription painkillers and heroin. Despite the lack of a reported overdose death, there is an LD50 for THC, apparently. The LD50 is the dose of a substance that is required to induce death in 50% of the animals in a toxicology study. I've never actually read the study or the paper that demonstrated this because it was published in 1974 and I haven't been able to get my hands on it, but it is referenced on the THC Wikipedia page. And it says that the LD50 of THC is 1,270 milligrams per kilogram. And the lower the LD50, the more toxic the substance. If we compare the LD50 of THC to aspirin, both administered orally, we can see that THC may be more than 6 times safer than aspirin, which is considered pretty safe. If we compare it to heroin, heroin is roughly 10 times more dangerous than oral aspirin, but it's not a totally fair comparison because they're using different methods of administration. I weigh 170 pounds, and there are, according to the state of California, only 100 milligrams of THC allowable in a brownie. My LD50 is 100,000 milligrams of THC, so I would need to consume 1,000 brownies in one sitting in order to raise the risk or the probability of death to 50%. And that is a lot of brownies. A systematic review of studies reporting adverse effects was published in 2008, and it found that almost 97% of all adverse effects were considered not serious. These non-serious adverse effects are largely dose and method of administration dependent. They include sedation, deficits in short-term memory and concentration, dizziness, euphoria or dysphoria depending upon a person's perception, side effects or adverse effects like cough, sputum production, and irritated airways would only occur with inhalation as a method of administration, for example. The most common adverse effect of THC is dose-related tachycardia, which is a good adverse effect to disclose to your patients during the informed consent conversation, because oftentimes that is enough to make them feel uncomfortable and spin them out into a mini panic attack situation where they feel like something very bad is happening to their heart and want to go to the emergency department. The methods of administration can be divided into 3 primary categories: inhalation, oral, and dermal administration. And patients may very well use more than one method of administration depending upon their clinical situation. The 2 primary benefits of inhalation are the rapid onset of effect and then the ability to regulate dosing based upon how you feel after a given dose. These compounds permeate the capillaries in the lungs. They pass into general circulation very quickly through the pulmonary arteries, and they cross the blood-brain barrier, and you're able to feel the subjective effects right away. So that could give you some feedback as to whether you might want to take another dose. The primary benefits of ingestion are the duration of the effect as opposed to the onset. It's longer. And the 11-hydroxy effect, which is a metabolite of THC produced during first-pass metabolism in the liver, and we'll talk about that. Oral absorption is said to produce an onset of effect that is somewhere in between inhalation and enteric absorption or ingestion. And I actually haven't seen good data on this, and my patients don't actually report that there is a real meaningful difference between ingestion and oral administration in terms of the onset of effect. But topical products may be adequate for things like cuts and scrapes, rashes, and joint pain. Without a carrier to drive the compounds deeper into the tissue, these products do not lead to impairment. The patient is highly unlikely to fail a drug test. Transdermal products, on the other hand, typically do have a carrier, and they are applied to highly vascular areas on the skin in order to initiate a systemic effect. And so with transdermal products, we are more likely to experience impairment, and the risk of failing a drug test is real with a systemic effect through a transdermal product. In the next few slides, we are going to look at the pharmacokinetics of inhalation versus ingestion. In the graph on the upper left, we are looking at levels of delta-9 THC in red and levels of 11-hydroxy-THC in orange in the blood after inhaling a joint or smoking a joint containing 15.8 milligrams of THC. You can see that the concentration of delta-9 THC in the blood spikes up to 80 nanograms per milliliter in the first 10 minutes, and that the curve for 11-hydroxy-THC is obviously much lower, and it peaks after the peak for delta-9 THC. In the graph on the right, we are looking at the same analytes after ingestion of a cookie containing 20 milligrams of THC. And we can see that the concentration of delta-9 THC actually is pretty low. It doesn't really have that same spiking profile, and that the levels of 11-hydroxy-THC are actually greater for most of this than delta-9. In this other graph, really at no point was 11-hydroxy greater than delta-9. This profile is indicative of ingested cannabis. This profile is indicative of inhaled cannabis. It's interesting to note that the subjective experience of being high is not well correlated with blood levels of delta-9 THC. In the graph on the left, we are looking at levels of delta-9 THC after inhalation and subjects' subjectively reported experience of being high, which is in orange here. Notice that the subjects are feeling higher as blood levels of THC decrease. In the graph on the right, we are looking at levels after ingestion of 20 milligrams of THC. The subjective experience of being high, here in orange, is rising up to 180 minutes after ingestion, whereas levels of delta-9 THC are relatively flat during that period of time. This slide does not include 11-hydroxy-THC, which is more potent than delta-9, and this is the metabolite that is largely responsible for the differences in effects when cannabis is ingested. This slide describes the proportion of delta-9 and 11-hydroxy-THC when cannabis is inhaled versus ingested. On the left here, we have the profile when inhaled, and on the right, we have the profile when ingested. These two are basically in a 1-to-1 ratio with one another during ingestion, but 11-hydroxy-THC is up to 7 times more potent than delta-9. And as a result, this ingested effect is usually an effect that's mediated by this particular metabolite, whereas the inhaled effect is mediated by delta-9 THC. There are some 100 metabolites of CBD that have been identified to date, but we don't know very much about them. The biological activity has not really been studied. Oftentimes you'll hear people refer to CBD in plural form, CBDs. And it's true, there are 4 different homologs of CBD with 4 different side chains, and the pentyl side chain is the molecule that we refer to as CBD. I don't think when people are referring to CBDs in plural, they're thinking about these other side chains. I think it's actually just a misunderstanding. In this slide, we're looking at 4 different volunteers: 836, 835, 837, and 846. And each of these subjects ingested a capsule containing 10 milligrams of THC and 5.4 milligrams of CBD. Let's look at the difference in the Cmax, the maximum concentration of delta-9 THC in volunteer 846, which is at 10 nanograms per milliliter, as compared to volunteer 835, which is 2.5 nanograms per milliliter. This is a difference of 4. That's 300%. Which one of these is your patient? Because in one situation, you may have underdosed the individual, and in another situation, you may have overdosed them. The important point here is there's so much biological variability that the pharmacokinetics can lead to a wide disparity between the dose that is administered and the dose that is actually received. A couple last points on this particular study. When CBD and THC were administered together, it led to greater bioavailability of THC than when THC was administered alone. So there's a different type of synergy there. That's a pharmacokinetic synergy. And then also, when THC was administered alone, the maximum concentration for the highest subject, or the highest concentration, was 8 times greater than the lowest concentration. That's, again, a huge disparity. Now I want to talk about the pharmacodynamic synergy. Here is a phase 3 clinical trial for Sativex, which is a drug that is not being evaluated currently by the FDA, but is approved in over 30 countries around the world. Sativex is an extract of cannabinoids and terpenes from marijuana. It's formulated to have a 1-to-1 ratio between CBD and THC. It was developed by the same company that developed Epidiolex. This is a randomized, double-blind, placebo-controlled study in patients with intractable cancer pain. And the outcome here is the percentage of patients experiencing a decrease in pain at various levels: 10% decrease, 30% decrease, and a 50% decrease over here on the far right. Notice that just over 20% of subjects experienced a 30 or more percent decrease in their pain when using THC, while 40% experienced that 30% decrease when they took THC and CBD in a 1-to-1 ratio with the same dose of THC. So here what we're seeing is some enhanced or some additional pain relief. Perhaps this is from the CBD itself. Perhaps this is from the synergy between the CBD and the THC. We don't actually know, because there was no CBD arm in this study that we could evaluate on its own to see what the effect of CBD alone might have been. In addition to clinical efficacy, we know from other studies that co-administration of CBD with THC may attenuate some of the adverse effects of THC. Now, there is some substantiation for this claim. I think we need more research because there's other data that refutes it. But the idea perhaps is that this effect is being mediated through the allosteric modulation of the CB1 receptor by CBD. And as a result, the change in shape leads to different pharmacodynamics and fewer adverse effects. It could also be due to the fact that CBD inhibits the metabolic enzymes in the liver that break down or degrade THC. And as a result, THC is not being converted to 11-hydroxy-THC at the same rate because CBD is inhibiting that conversion. And as a result, perhaps they're not experiencing as much of a potent 11-hydroxy-THC effect, which may have a higher adverse effect profile. Now let's talk about the dose-response relationship. This is a randomized, double-blind, placebo-controlled study. It was a crossover study among patients with neuropathic pain. The subjects were given different potencies of marijuana to smoke. They were given a 0% potency, 2.5, 6, and 9.4%. All of these potencies would be considered very low based upon what's available today at cannabis dispensaries and on the illicit market. The outcome was the average pain intensity score over 5 days, and the author stated that the average daily pain intensity was significantly lower on 9.4% THC cannabis as compared to 0% THC. And the absolute reduction in pain scores is frankly quite low. It's 0.7 on an 11-point scale. Before we look at a comparison of the different potencies in that clinical trial, let's quickly look at the difference in general between potency and dose, and the difference also between the dose administered and the dose received. So participants in this study smoked a single 25 milligram dose, in quotes, of cannabis flower through a pipe 3 times daily for the first 5 days of each cycle. And it was a crossover design, so they tried every different potency, including the placebo. So if we assume a different potency, 9.4% versus 2.5%, you could see that the dose that's actually delivered is very different. And in this clinical trial, this is a very low dose, both of these. If we assume a standard absorption, let's just say 35%, which would be between ingestion and inhalation, let's say, we can also see, therefore, that the dose that's actually received is different. If we change the method of administration and say the 9.4% potency was inhaled and the 2.5% potency was ingested, for example, we can see that the dose received is actually even more different than the dose delivered. So potency and method of administration are very important in trying to understand dose. Okay, now back to this study. This is table 2 from the paper. These are changes in mean daily pain scores across the different potencies, which, as we just saw, equates to different dosages. The first column compares the change in mean pain scores for each potency as compared to placebo. So 2.5 compared to placebo, 6 compared to placebo, etc. The 2.5 potency led to a reduction of 0.13 points on an 11-point scale for pain as compared to placebo. The 6% potency led to a lesser reduction than the 2.5% potency, while the 9.4% potency led to a greater reduction than both. Still, from an absolute perspective, this is a pretty small reduction in pain, less than a point. Notice that this is not a linear trend. The second column compares the potencies to the 2.5% potency. Basically, no difference between 6% and 2.5%. And then a small, albeit not statistically significant difference between the 9% or the 9.4%, excuse me, and the 2.5% potency. And then finally, in the third column, the comparison is between the 9.4% and the 6% potency. And again, a small but not statistically significant reduction in pain scores. So these confidence intervals, for the most part, are pretty wide. Some of them cross 0. And overall, these changes in pain or the reductions in pain are very small. That's a different conversation. The point of this really is about the absence of a clear dose-response relationship. And there are other studies like this. This is just one of the studies that looks at different doses or different potencies. This study was a double-blind, placebo-controlled study investigating the anxiolytic effects of CBD in patients with social anxiety. There were 5 different groups. There was a placebo, a clonazepam, and then 3 groups of CBD at different dosages, 100, 300, and 900 milligrams. And notice that the 300 milligram dose of CBD led to greater reductions in post-stress anxiety than both the 100 and the 900 milligram doses. And this difference was statistically significant when compared to both the placebo and the clonazepam. You'll see this referred to as a bell-shaped curve, where the greatest effects are often in the middle of the dosing range, not at the higher end. And this obviously is looking specifically at CBD and specifically at isolated CBD. This preclinical study in rodents is a linchpin in the argument that whole plant extracts have different pharmacodynamics than isolated cannabinoids. The comparison of these two graphs shows a very different story between the dose-response relationship. The vertical axis is PAW withdrawal threshold in grams. This is the mechanical force that is required to motivate the rodent to withdraw its inflamed paw after being poked with a filament. And the paw is inflamed because the investigators injected capsaicin into the paw. So paw retraction in this study was used as a proxy for the anti-nociceptive effects of CBD. The higher the force that is required to induce a retraction, the greater the anti-nociceptive effects. So on the left, we have the CBD isolate, and in the graph on the right, we have the whole plant extract. Notice the dose response curve for the CBD isolate. The threshold for paw retraction actually decreases over time. Now, notice the curve on the right. If we're looking at 6 hours in particular after administration of CBD, you'll see this basically linear dose-response relationship where the higher the dose, the greater the force required to motivate the rodent to withdraw its paw. I think it's important to note here that the doses are still very high, 150 milligrams, 50 milligrams per kilogram. And the outcome for analgesia here in the study is not very specific, especially if we are trying to extrapolate to human beings. But this is something that gives us a sense for differences in the dose-response relationship between an isolate and a whole plant extract, and then we can see this linear curve. As a side note, this study also shows decreases in inflammation. This study is entitled Overcoming the Bell-Shaped Dose Response of Cannabidiol. And I think it's fair to say that we actually don't really know the shape of the dose-response curve yet. This study, as I mentioned, is often cited to support the argument that a whole plant extract has different pharmacodynamics than an isolate. There are obviously differences between rodents and humans. There are differences between methods of administration, and there may be differences between isolated versus whole plant extracts. So it's important to understand that this question is still outstanding. There are 5 primary contraindications to consider when using cannabis in clinical practice, and these contraindications are specifically related to using THC and THC-containing products. Some are absolute contraindications, while others are more relative. You will find, I think, quite a bit of variability in how clinicians consider these different contraindications. My opinion is that THC should be contraindicated during pregnancy and lactation. Now, there is evidence that supports this, but there's also evidence that refutes it. The evidence that THC can negatively affect neurological development is enough for me to want to exert a lot of caution in that particular clinical circumstance. Obviously, there are scenarios where you may need to negotiate between various potentially harmful interventions, and those scenarios should be considered on a case-by-case basis. It's also important to understand a given individual's mental health history. Associations between THC use and schizophrenia have been reported in epidemiological studies, but the relationship is not causal. There are some case studies more recently where adolescents experienced psychotic episodes after dabbing or inhaling very high doses of THC. The dose-related tachycardia is the most common adverse effect of THC use. THC and CBD both lower blood pressure, and as a result, the heart increases its rate and its contractility in order to maintain cardiac output. So the drop in blood pressure that may be associated with using these cannabinoids could potentially induce tachycardia that might be harmful for patients with unstable cardiac conditions. There are studies that indicate that smoked cannabis may improve lung function in individuals with asthma and COPD, but despite these studies, I think it's probably a good idea to avoid inhalation in patients that have compromised respiratory function. THC use among adolescents and among children should be monitored to avoid abuse. The effects on brain development are associated with chronic use, not intermittent use and not high dose use. With regard to CBD specifically, Epidiolex, which was mentioned before, was placed on Schedule 5 by the DEA in 2018 in the fall, which means that it has a very low potential for abuse. In 2017, the World Health Organization published a preliminary report stating that CBD had a very favorable safety profile. So I think there are fewer contraindications for using CBD than for using THC. As far as herb-drug or drug-drug interactions are concerned, inhibitors of the cytochrome P450 enzyme system will inhibit the metabolism of THC and CBD, while inducers will induce it. I think it's estimated that up to 60% of all drugs, prescription and illicit, are metabolized by that cytochrome P450 enzyme complex. One pharmacokinetic study found that the antifungal drug ketoconazole, which is an inhibitor, increased the plasma concentration of CBD by about twofold, while the antibiotic rifampin, which is an inducer, reduced plasma concentration of CBD by approximately 50 to 60%. In a different study, omeprazole, the proton pump inhibitor, which is a modest inhibitor of cytochrome P450 2C19, did not alter the plasma concentration of CBD, and 2C19 is one of the subunits that metabolizes CBD. Clinically significant interactions that are caused by THC or CBD are fairly rare. We don't have a ton of data on this, but the data that we do have comes from FDA-approved studies looking at synthetic isolated THC, which are the drugs that I mentioned in the beginning of the presentation, and from the clinical trials looking at Epidiolex, which is the CBD extract that was approved to treat the pediatric seizure disorders. It's possible that interactions are different for isolates than for whole plant extracts, and both THC and CBD are inhibitors of CYP450. CBD is a more potent inhibitor. CBD actually is a competitive inhibitor in that it will displace other ligands that are bound to the site of enzymatic activity. CBD is also a more potent inhibitor than the grapefruit compound bergaptan. There's a variety of different components or compounds that are in grapefruit that have been shown to inhibit the CYP450 complex, and CBD is a more potent inhibitor than bergaptan. In a 2013 clinical trial looking at Sativex, which is GW Pharmaceuticals' 1-to-1 CBD to THC liquid extract from marijuana, which was not approved in the US because the application was not submitted, but is approved in other countries, found no interactions when dosing up to 40 milligrams of CBD. And that also, because it's a 1-to-1, means that there were 40 milligrams of THC also administered without a significant drug interaction. In the clinical trials investigating Epidiolex, the levels of other anti-seizure medications were monitored very carefully. The metabolism of the anticonvulsant drug clobazam was really the only drug that was inhibited enough to produce a clinically significant interaction. They noted that the dose of clobazam had to be reduced in a number of patients because of this interaction. There was a published case study of an interaction between CBD and warfarin, but the dose of CBD in that case study was well into the hundreds of milligrams. As an inhibitor, generally speaking, but specifically with regard to warfarin, CBD leads to that inhibition leads to higher blood levels, a higher plasma concentration of whatever compound is being inhibited for a longer period of time. And so as a result, the patient is more likely to experience either greater efficacy, longer efficacy, or perhaps a higher rate or greater severity of adverse effects that would be mediated by higher levels of the drug in the blood for a longer period of time. I often say that THC is not a good drug, and that's because it has a very narrow therapeutic window. The difference between the minimum effective dose and the maximum tolerable dose is very small. And it happens to have this side effects of intoxication, which can lead to impairment. And this is an adverse effect for most patients, but not all patients, or sometimes it's an adverse effect at a certain time of day, but not an adverse effect at a different time of day, like nighttime, for example. I talk to patients about this concept of the minimum psychoactive dose, and that is the minimum dose of THC in milligrams that results in a noticeable change in perception. It's a measure of intoxication or THC-like effects. And the goal is to really understand how the patient feels about these intoxicating effects. Do they feel uncomfortable? Is it dysphoria? Do they feel comfortable? Is it euphoria? Because how your patient is able to tolerate those effects gives you a lot of information about the dosing range that you can play with when you're trying to focus on symptom control. I try not to use the word psychoactivity when I'm talking with clinicians or researchers because that word is often used incorrectly in clinical practice and in research and in the media when referring to the absence of intoxication when using CBD. So people say that CBD is not psychoactive because it doesn't get you high. But we should be clear, CBD is absolutely psychoactive. If it treats anxiety and depression and PTSD, if it treats pain, if it interacts with serotonin receptors and GABA receptors, which it does, if we lead patients to expect some sort of change in how they feel, their perceptions of stress or their perceptions of pain, well, we can't say it's not psychoactive. So this minimum psychoactive dose is something that patients will latch onto. The word psychoactivity seems to be something that is really attractive to people, but it's being used incorrectly, at least in my opinion, in a lot of circles. Once the minimum psychoactive dose is determined, you now have a baseline for beginning a dosing regimen that is tailored towards symptom reduction or clinical efficacy. The way that I approach it, initially, we're trying to understand tolerability. And so the dosing in the very beginning is to start with a very low dose so that they don't feel uncomfortable. And then once you understand what level might lead to discomfort, you know where you can start when it comes to the primary objectives, which are symptom control. And the pharmacokinetics, the pharmacodynamics, as we saw many slides ago, are specific to your patient, and you don't know what they are at the initial visit. And so that's why using these compounds in clinical practice requires a lot of time and effort and education, because you really have to start at a different place for each patient. This chart was taken from one of the phase 3 clinical trials investigating Sativex. It shows 3 different groups in the clinical trial: a THC group, a placebo group, and a THC and CBD group. This is the Sativex group. One spray of Sativex is 100 microliters, and it contains 2.7 milligrams of THC and 2.5 milligrams of CBD, so basically a 1-to-1 ratio. The vertical axis here is the mean number of sprays per day, and the horizontal axis is the number of days. The most important point here is that there's this gradual dose titration phase over the first 7 days of the study. And these individuals were self-titrating, and these are means in each group. But you can see that on their own, they started at a certain level with some guidance from the investigators, but worked their way up each day until about the 7th day, where most of these plateaued in terms of the amount that they were using or the total dose of THC and CBD, which on average is about 30 milligrams of each per day. The minimum psychoactive dose that we talked about in the previous slide is focused on THC, not CBD, because THC is so much more potent. A very small amount of THC can have very meaningful clinical effects for your patients, and even a moderate dose of THC can have potentially deleterious or harmful or at least stressful consequences for your patient. When it comes to using THC, it can be a bit of a balancing act between symptom relief and impairment. And many patients are able to achieve adequate symptom relief without experiencing any impairment whatsoever, but others may need to tolerate some degree of impairment in order to achieve an adequate degree of symptom relief. And oftentimes, patients will take a different dose at one time of day, perhaps in the morning, and achieve a certain degree of symptom control, let's say perhaps 30% pain relief. And then maybe they'll take a higher dose in the late afternoon or in the evening where it's okay for them to feel a little bit of impairment. And as a result, they'll also get a corresponding incremental decrease in their symptoms. So maybe they take a higher dose in the afternoon and the evening, and instead of getting 30% pain relief, they can experience 50% pain relief. To some extent, we can modulate this impairment by administering CBD either concurrently or preemptively, or we can use multiple and different methods of administration at different times of day in order to get the effects, both desired and adverse, that allow a person to navigate their daily activities. There are also products that include the acid forms of these cannabinoids. THCA and CBDA do not induce any type of intoxication. I guess I'm specifically referring to THCA, and are also demonstrated to be anti-inflammatory. And then there are other cannabinoids like CBN, CBG, and delta-8 THC that can also be useful. A few words about the challenges of using cannabis or cannabinoids in clinical practice. First, there is a lot of fear, uncertainty, and misinformation among patients. They are generally curious, but they're also cautious and they're confused. Many patients are already experimenting on their own with products that they purchased from licensed or unlicensed sources, products that have labels that lack important information a lot of the time, like the dose in milligrams. Almost universally, patients don't want to get high, and they're now being told that the way to avoid getting high, the way that they can use medical cannabis without getting high, is by using CBD, which is fine because CBD does a lot, but it doesn't do everything that THC does. It doesn't stimulate appetite. It doesn't relax muscles. It doesn't treat neuropathic pain or reduce nausea the way that THC does. Tolerability can be a big limitation for some patients, and that can be a challenge. Patients that are intolerant to the intoxicating effects of THC may not be good candidates for it. And due to federal prohibition, we lack dosing guidelines, we lack standards of care for using cannabis and cannabinoids in clinical practice. The information we have for the synthetics is not easily generalizable to medical cannabis products that are available either legally or illegally in most states. Also, we are unclear about the dose response curve. Is it linear for a whole plant extract, but bell-shaped for an isolated cannabinoid? Does it depend on the individual's pharmacokinetics? Maybe it depends on the method of administration. There's a lot of questions that matter that still need to be answered. Product availability is another important issue, even in states with relatively mature and regulated medical and or recreational cannabis marketplaces. Many products are here today and then gone tomorrow, and the patient is left not knowing what to replace it with. Cost is obviously a challenge. These products are not covered by insurance. They're expensive. Even hemp-derived CBD products can be expensive depending upon the dose. And finally, the regulatory environment is constantly changing. Hemp-derived CBD products are available today over the counter and online as if they were dietary supplements. Yet the FDA explicitly states that they are not dietary supplements, nor for that matter are they intended to be used to treat, diagnose, mitigate, or prevent disease, unless we're talking about Epidiolex, which requires a prescription and is only approved to treat 2 rare pediatric seizure disorders. But despite all of that, that's exactly why and how people are using these products to treat symptoms and to treat disease, yet they're not approved to do that. Thank you for your time and attention. Please feel free to reach out to me if you have any questions. My contact information is below.